Okay, let's unpack this. Imagine a highly sophisticated, self-repairing machine, much like our own bodies, right?
Mm-hmm.
This machine is designed with an incredible emergency brake, a mechanism that, in its prime, can instantly stop disaster.
The lifesaver.
Exactly, a lifesaver, a critical defense. But what if, as this machine aged, that very same emergency brake didn't just engage when needed, but started getting stuck on?
Uh-huh. Stuck on.
Yeah, like its persistent engagement, even when there wasn't an immediate threat, actually led to chronic wear and tear, and eventually driving the very decline it was initially meant to prevent.
That's a powerful analogy.
Well, today we're diving deep into cellular senescence, a fascinating biological phenomenon that truly embodies this paradox. It's often described as the double-edged sword of aging.
Right.
It's a fundamental stress response program where cells permanently stop dividing, and in its acute phase, it acts as a crucial defense mechanism against something truly formidable: cancer.
What's truly fascinating here is precisely that dual nature, this profound biological irony. In its initial acute phase, senescence is indeed a protective guardian, a cellular hero, you could say.
Right.
It's an elegant system of cellular self-defense where a cell, upon detecting significant damage or maybe dangerous changes, it deliberately shuts down its own ability to proliferate.
Stops it from becoming cancerous.
Exactly, preventing it from turning malignant. But simultaneously, it actively calls for help, sending out specific signals to ensure the damage is contained, repaired, or the problematic cell is cleared out.
Okay.
It's a beautifully orchestrated short-term response, vital for maintaining cellular integrity and, the health of the whole organism.
Yeah.
But, and here's the twist, as these senescent cells accumulate, particularly in our later years, they transition.
From heroes to
From protective allies to persistent drivers of chronic inflammation and tissue degeneration. And this immediately raises a profound scientific and honestly philosophical challenge for us. Can we intervene? Can we selectively target these senescent cells, or maybe just their troublesome secretions, without undermining the very, very important benefits they offer in early life?
That is the million-dollar question, isn't it? To tackle this intricate puzzle, we're going to embark on a deep dive through the latest research. First, we'll meticulously dissect exactly what cellular senescence is.
Lay the groundwork.
Exactly, exploring its initial, undeniably beneficial roles. Then we'll pivot to understanding how and why it becomes problematic as we age. We'll pinpoint the specific cellular triggers that force a cell into the senescent state.
The why now triggers.
Right. And the core biological pathways that enforce that irreversible growth arrest. From there, we'll delve into the notorious SASP, the senescence-associated secretory phenotype.
Ah, yes, the SASP.
This powerful, complex cocktail of molecules these cells continuously release. We'll examine how it can both heal and harm, depending on context and duration.
Crucial distinction.
And finally, we'll look at groundbreaking evidence from landmark mouse studies that didn't just correlate these cells with aging, but actually causally linked them to age-related decline. And this offers tantalizing possibilities for intervention in human health. So, let's start at the very beginning, defining this pivotal cellular state. Cellular senescence, at its core, is a cellular stress response program. Now, what does that really mean for a cell?
Well, it means that when a cell is faced with certain kinds of severe, unmanageable damage or profound stress, it doesn't just die. That's apoptosis.
Right, it's not just self-destruction.
No. And it doesn't continue dividing recklessly either, which would be cancer. Instead, it initiates a deliberate, highly organized genetic program to permanently cease dividing.
Permanently, not just a pause.
Exactly. This isn't a temporary pause like hitting a pause button on your music. It's an irreversible growth arrest. Think of it like throwing a wrench into the gears of the cell division machinery and locking it down permanently.
Wow.
And this permanent cessation of proliferation is what makes it such an effective emergency brake against potential catastrophe.
So let's talk about that catastrophe. How does it stop it?
Well, consider a scenario where a cell has accumulated too much DNA damage, maybe from chronic exposure to, say, UV light or environmental toxins, or even just errors during the incredibly complex process of DNA replication itself.
Which happens all the time, presumably.
It does, yes. And under normal circumstances, such a damaged cell, if it continued to divide, could proliferate uncontrollably. It would pass on its errors, potentially forming what we call a nascent tumor, the very beginning of cancer.
Okay.
Senescence steps in at that critical juncture, like a physical brake, engaged with tremendous force. It locks that cell in place, preventing any further division and proliferation.
So it nips it in the bud.
Precisely. It acts as a primary, fundamental tumor suppressive mechanism that protects the organism, especially in its younger, more vulnerable years. It effectively puts a stop to the potential for malignancy before it can even truly begin.
So it's a profound anti-cancer mechanism, a cellular sentinel, really. But you mentioned earlier that it's more than just preventing cancer. How does this emergency brake benefit a young, healthy organism in other ways?
Oh, it's absolutely crucial for other healthy physiological processes, too. Beyond tumor suppression, senescent cells play significant beneficial roles in healthy young organisms, particularly in tissue remodeling.
Tissue remodeling, like sculpting.
Sort of like sculpting, yes. Think of the human body not as a static structure, but as a constantly evolving, self-sculpting entity. Tissues are continuously being refined, restructured, maintained throughout life, right from embryonic development.
Building the body plan.
Exactly. And through puberty, all the way to wound healing in adulthood. Senescent cells aid in this sculpting process. For instance, in embryonic development, they're involved in shaping organs, guiding the removal of unnecessary cells or structures.
Huh. I never thought of that.
And in adults, they facilitate the healthy turnover and reorganization of tissues, ensuring their optimal form and function. They're like microscopic architects almost, overseeing the demolition of old structures and preparing for new ones, ensuring the integrity and adaptability of our tissues.
That's a fascinating, almost artistic role for something we typically associate with decline. And I understand they also play a vital, acute role in wound healing.
Precisely. When you experience an injury, whether it's a simple cut on your skin or maybe more significant internal damage to an organ, senescent cells transiently appear right at the site of injury.
They rush to the scene.
In a manner of speaking. They don't simply arrive to shut down. Their presence is a calculated, orchestrated part of the immediate repair process. They are integral to the acute beneficial response that allows tissues to mend and recover.
So their appearance is actually a good sign initially.
Initially, yes. It's a sign of health and active repair, a crucial component of the body's natural defense and recovery system.
Okay, so how do they do all this? What's the mechanism? You mentioned growth arrest.
Right. The core mechanism through which senescent cells achieve these multifaceted effects is twofold. First, as we've discussed, there's the irreversible growth arrest, that permanent stop to cell division. That's the defining feature, what marks them as senescent.
Got it.
But it's not just that they stop dividing, they also become highly active secretors. And this brings us to the senescence-associated secretory phenotype, or SSP.
Okay.
The SEP is a truly critical component of how senescent cells exert their profound effects, whether those effects turn out to be beneficial or detrimental down the line. It's a complex, potent cocktail of dozens, maybe even hundreds, of distinct signaling molecules that these cells actively spew out into their surroundings.
Phew. Sounds messy.
Well, it's a very active process. They essentially transform into little cellular factories, constantly releasing these factors that profoundly influence their microwave environment, affecting neighboring cells and sometimes even distant tissues.
So it's like they're not just stopping their own activity, but they're also sending out a complex, coded broadcast, a constant stream of signals into their neighborhood, profoundly altering the local tissue ecosystem. This complex mix must have diverse impacts.
Absolutely. In the short term, this SAFP provides significant benefits. It acts as a crucial communication hub. For instance, SASP factors are exceptionally effective at immune cell recruitment.
Calling in the cleanup crew.
Exactly.
Yeah.
They act like chemical flares or a strong magnetic pull, attracting various immune cells, macrophages, natural killer cells, T lymphocytes, a full cellular cleanup crew right to the site of damage. This is essential for clearing away dead cells, cellular debris, or even pathogens. Crucial for maintaining tissue integrity and preventing infection.
Makes sense.
Furthermore, the SASP directly contributes to promoting repair following injury. The molecules released can stimulate neighboring healthy cells to proliferate and differentiate, aiding in the regeneration of damaged tissue. They can even guide the migration and activity of crucial stem or progenitor cells, ensuring they arrive at the right place at the right time to rebuild.
Wow, very sophisticated.
And critically, this recruited immune surveillance, guided by the SASP, plays a direct role in the suppression of nascent tumors. Immune cells, drawn by SASP signals, can identify and eliminate potentially cancerous cells that might have slipped past other defenses before they can establish a full-blown malignancy.
So it's double protection. The cell stops itself and it calls in backup to make sure.
Precisely. It's a beautifully orchestrated short-term response, an elegant system of cellular self-defense. The cell detects damage, shuts down its own proliferation to prevent malignancy, and then calls for help, via the SASP to ensure the damage is contained or repaired.
That sounds like an incredibly efficient, even elegant system. It's truly a cellular hero in its early stages. But you mentioned earlier it's a two-edged sword. So what's the other edge? When does this beneficial acute SASP response become problematic? What's the tipping point?
That's the core paradox we're grappling with. While acutely beneficial and transient in healthy young tissues, senescent cells, well, they accumulate significantly as we age.
They stick around.
They don't get cleared as efficiently by the immune system, which itself can become less robust with age, or maybe they simply become too numerous to be efficiently removed. This leads to their prolonged persistence in various tissues throughout the body.
Okay.
And this persistence means that the beneficial acute SASP, which was designed for temporary action, becomes chronic. Instead of a temporary cleanup crew signal, you have a permanent low-grade inflammatory state.
Like a fire alarm that never turns off.
Exactly like that. This chronic SSP creates a hostile, pervasive, pro-inflammatory environment in tissues. Scientists often call it a milieu, a continuous, unfavorable surrounding condition, a low-level assault that just doesn't let up.
And that can't be good long-term.
It's definitely not benign. This chronic inflammation actively interferes with the normal, healthy functioning of tissues and organs, leading to widespread dysfunction over time.
So what's the cost to us, the organism?
The organismal cost of this chronic state is substantial. The sustained low-grade sterile inflammation driven by chronic SSP is a major contributor to what's been termed inflammaging.
Inflammation driving aging.
Precisely. It's now recognized as a key hallmark of aging itself. This chronic SISP leads directly to widespread tissue degeneration, the breakdown, deterioration, and dysfunction of normal tissue structure and function. We're talking about direct, persistent damage at the cellular and tissue level, undermining the very foundation of our organs.
And this shows up as specific diseases.
It manifests in the specific aging pathologies we unfortunately see all around us. Conditions like insulin resistance, a precursor to type 2 diabetes, atherosclerosis, the hardening of arteries causing heart disease and stroke, osteoarthritis, the painful, debilitating degeneration of joints, and various forms of neurodegeneration impacting brain function and leading to conditions like Alzheimer's and Parkinson's.
Wow, that's a major list.
It is. These are all common debilitating age-related diseases that are now understood to be significantly driven, at least in part, by the accumulation of senescent cells and their chronic SASP.
So what we're learning is that a mechanism that starts as a lifesaver, a cellular hero designed for protection and repair with time, transforms into a silent saboteur, quietly undermining our health from within, piece by piece. This truly brings us back to the central question you posed earlier, this profound paradox of program versus pathology.
Exactly. The core question for researchers and clinicians, therefore, becomes this. Can we selectively target these senescent cells or their problematic SAP to treat, maybe even prevent, these age-related diseases without compromising their essential protective physiological benefits?
The tightrope walk.
It's a major challenge, and this defines the rapidly emerging and incredibly exciting field of xenotherapeutics. These are drugs or interventions specifically designed to alter the course of senescence.
Xenotherapeutics, okay.
The field is broadly pursuing two main strategic avenues, either to clear these senescent cells entirely, literally eliminating them from the body using drugs often called senolytics, or to calm their troublesome secretions, essentially silencing their chronic SAP without necessarily killing the cell. That strategy uses xenomorphics.
Clear or calm. Got it. And the goal is?
The overarching goal here isn't simply to extend chronological lifespan, make people live longer in the state of frailty. Critically, the aim is to extend healthspan.
Healthspan, the healthy years.
Exactly. The period of life spent in good health, free from debilitating disease and disability. It's about living better, for longer.
It's an ambitious goal, aiming not just for more years, but more healthy years. So just to make sure we're all tracking here. We started with senescence as this crucial anti-cancer emergency brake.
Right, with irreversible growth arrest.
And its acute beneficial SSP helps with tissue repair and remodeling.
Essential in youth.
But then, tragically, it transforms into a chronic issue. These cells accumulate, the SSP becomes persistent,
creating chronic inflammation, the inflammaging,
which drives a significant portion of aging pathologies, fueling everything from insulin resistance to neurodegeneration.
Correct.
And the promise of xenotherapeutics is to specifically intervene either by clearing or calming these cells to extend that vital health span.
That's the hope and the focus of intense research right now. It really underscores how a process evolved for immediate survival can become a liability later in life.
Okay, so we understand what cellular senescence is and what it does, both the good and the bad. But what actually pushes a cell over the edge into this growth-arrested senescent state? You mentioned stress.
Right. It turns out there's not just one switch, not one single event. It's more like a complex network of different cellular stressors that can all trigger this program. They act like different alarm signals, telling a cell, okay, halt, it's time to stop dividing and enter this protective state.
So multiple roads lead to senescence.
Indeed. We're talking about multiple distinct roads leading to the same critical cellular state, which really reflects the robustness of this defense mechanism. One of the most classic and well-studied triggers is called replicative senescence.
Replicative senescence due to replication.
Exactly. This occurs because of the progressive shortening of telomeres. Now imagine your chromosomes carrying all your genetic info, like shoelaces.
Okay, I like the shoelace analogy.
Telomeres are the protective plastic tips at the ends of those shoelaces. Aglets, I think they're called.
Right.
They're specifically designed to prevent the chromosomes from fraying or from, aberrantly fusing with other chromosomes, which would be disastrous.
Makes sense. Protective cap.
Protective caps.
Yeah.
But here's the thing. Every time a cell divides, the cellular machinery that copies the DNA can't quite replicate the very, very ends of the chromosomes perfectly. So a small piece of these telomeres is lost.
With every single division.
With every division. It's a natural consequence of DNA replication. So after repeated cell divisions, typically around 50 to 70 divisions for many human somatic cells, a phenomenon known as the Hayflick limit, the telomeres become critically short.
They wear down.
They wear down. And when they get too short, they are no longer recognized by the cell as normal protective chromosome ends. Instead, the cell's internal machinery perceives them as significant and irreparable DNA damage signals.
So short telomeres look like broken DNA.
Essentially, yes. And this triggers the senescence program. It acts almost like a built-in cellular clock mechanism, saying, okay, you've divided enough times, your genetic material is now potentially at risk, it's time to retire and stop dividing.
So replicative senescence is almost like a scheduled retirement based on a cell's division history. But what about sudden, unexpected damage? Does that also trigger senescence?
Absolutely. Telomere shortening isn't the only way in. Direct DNA damage can induce senescence at any age, even in young cells that haven't undergone many divisions at all.
Okay. Where does that damage come from?
It can originate from a variety of sources. It can be external, like radiation exposure, like ultraviolet UV radiation from the sun causing DNA lesions in your skin cells, or maybe ionizing radiation from medical treatments or the environment.
Right.
Or it can come from internal sources, such as reactive oxygen species, ROS.
ROS, the metabolic byproducts.
Exactly. ROS are highly reactive molecules, essentially harmful byproducts of normal cellular metabolism. Think of them like exhaust fumes from your car's engine. If these ROS build up, maybe due to stress or inefficient metabolism, or if there's significant oxidative stress, they can directly damage cellular components, including DNA, proteins, lipids.
All sorts of things.
All sorts of things. And this kind of direct genotoxic stress bypasses the need for telomere shortening and directly triggers the senescence program. The cell senses the danger and forces itself into arrest to prevent propagating a damaged genome.
So it's not just about how many times a cell has divided, but also the quality and integrity of its DNA throughout its lifespan. What other critical internal threats can trigger this protective shutdown?
Another really powerful trigger is the inappropriate activation of oncogenes.
Oncogenes, the cancer-promoting genes.
Precisely. Oncogenes are specific genes that, when mutated or hyperactive, have the dangerous potential to promote uncontrolled cell growth and ultimately cancer. A very well-known example is a ROS oncogene, but there are others like MYCOC or BRAF. When an oncogene like ROS becomes aberrantly active, maybe due to a mutation, it could drive cells into a dangerous hyperproliferative state. It forces them to divide uncontrollably and at an incredibly rapid pace.
The very beginning of cancer.
Exactly. This uncontrolled division is a hallmark of early cancer development. In this scenario, senescence acts as an essential emergency brake, halting this runaway process through a mechanism called oncogene-induced senescence, or OIS. It effectively prevents the formation of a full-blown tumor.
Another layer of cancer protection.
A critical one. And even something as fundamental as mitochondrial dysfunction can contribute. Mitochondria, as you know, are the powerhouses of the cell, generating most of its energy.
Right, ATP production.
When they malfunction, maybe due to damage or age. They can produce excessive ROS, leak mitochondrial DNA into the cell cytoplasm, or alter key metabolic ratios. All of these create significant cellular stress that pushes the cell towards a senescent state.
So telomeres, DNA damage, oncogenes, mitochondria, quite a few triggers. Do they all use different mechanisms to induce senescence?
That's what's crucial to understand. Despite these diverse triggers, whether it's telomere erosion, direct DNA damage, oncogene activation, or mitochondrial issues, they don't each activate a completely separate senescence pathway. Instead, they all seem to converge on a common set of critical cellular signaling networks.
Like different roads leading to the same central command center?
Exactly like that. And these central command pathways are primarily the P53 pathway and the P16-INK4A pathway. Both of these involve major tumor suppressor proteins.
P53 and P16.
The P53 protein, often called the guardian of the genome, is a master transcription factor. When it's activated by DNA damage or other stresses, P53 ramps up the expression of P21-CIP1, which is a powerful cell cycle inhibitor.
So P53 tells P21 to put the brakes on.
In essence, yes. And similarly, P16-INK4A is another key player. Its activation ultimately leads to the activation of RB, the retinoblastoma protein, which is another crucial tumor suppressor that controls cell cycle progression.
This is where the molecular mechanics become particularly elegant. So P21 and P16 are the direct molecular players, the enforcers, that actually halt the cell cycle and enforce this irreversible growth arrest.
Exactly. Think of P53 and P16 as master switches within the cell. When the cell detects severe stress from any of the triggers we've discussed, these master switches get flipped. P53, when activated, increases the production of P21, and P16, when activated, directly inhibits key enzymes called cyclin-dependent kinases, or CDKs, specifically CDK4 and CDK6.
CDKs, they drive the cell cycle forward.
They're the molecular engines, yes. They literally push the cell through its division cycle, particularly at the critical G1S checkpoint. That's the point where the cell commits to synthesizing new DNA and preparing for division.
A point of no return, almost.
Pretty much. By inhibiting these CDKs, P16 ensures that the RB protein remains in an active dephosphorylated state. The active RB protein then physically binds to and sequesters other proteins, like transcription factors called E2F, which are absolutely essential for activating genes required for cell division.
So RB holds back the go signal.
It does. So in essence, P21 and P16 directly put the brakes on the cell's ability to divide. It's a beautifully conserved molecular program, ensuring that the cell cycle is arrested and remains arrested, making the growth arrest irreversible.
So it's robust. Can a cell use either pathway?
Yes. It's important to note that cells can enter senescence via either pathway, or sometimes both simultaneously. It showcases the system's robustness and redundancy. For example, acute severe DNA damage typically stabilizes and activates P53 very quickly, which then leads to a rapid upregulation of P21, directly causing cell arrest.
Fast response to damage.
Right. In contrast, chronic mitogenic stress may be persistent signals encouraging cell division, or just the cumulative cellular stress associated with the process of aging itself that often progressively elevates P16 levels over time.
A slower, more cumulative trigger.
Exactly. This elevated P16 then persistently silences those cyclin-dependent kinases, thereby enforcing the R brake on proliferation more chronically.
And can we detect these pathways being active?
We can. Notably, the expression of both P16 and P21 often serves as molecular markers for the senescent state in cells, helping researchers identify these cells in tissues. And P16-INK4A, in particular, has emerged as a really broad and significant biomarker of senescent cells across various tissues and species.
Why P16 specifically?
Because its expression reliably rises with age in numerous rodent and human tissues. It's almost like a molecular clock ticking up. This makes it a powerful indicator of the overall senescent cell burden in an organism as it gets older. Think of it like a unique molecular signature, or maybe a fingerprint for senescent cells that just becomes increasingly prominent with the passage of time.
That sounds incredibly useful for research.
It absolutely is. And this detailed understanding of P16's role has led to truly ingenious experimental tools. For instance, scientists figured out how to use the P16 gene promoter.
The on switch for the P16 gene.
Right. That specific regulatory DNA sequence that controls when and where the P16 gene is turned on. They've cleverly utilized it in sophisticated transgenic mouse models. A famous example is the INK attack mice.
INK attack.
Okay.
What does that do?
Imagine scientists genetically engineering these mice so that the P16 promoter is precisely linked to a suicide gene.
A suicide gene. Wow.
Yeah. It means that when a cell becomes senescent and starts expressing P16 because the promoter gets activated, the suicide gene is also activated, effectively tagging the senescent cell with the molecular flag.
Marking it for destruction.
Exactly. Then, by administering a specific drug, usually a harmless chemical compound called a dimerizing agent, scientists can activate the suicide gene only in the tagged cells, leading to the selective and precise elimination of only those senescent cells from the mouse.
That's incredible. So they can specifically delete senescent cells.
Precisely. This technological breakthrough is absolutely crucial because it allowed researchers to move beyond just observing senescent cells to directly manipulating their presence. They could finally study the causal effects of their removal, which we'll definitely discuss in more detail later. It was a game changer.
Okay, so summing up this chapter, multiple roads lead to senescence, telomere shortening, DNA damage, oncogenes, mitochondrial problems.
Diverse stressors, yes.
But they all funnel through these common and highly conserved tumor suppressor pathways involving P53, P21, and P16-RB.
The central command.
These pathways are the cell's internal police force, enforcing the irreversible growth arrest.
Putting the brakes on hard.
And this cell-autonomous tumor suppression mechanism, where the cell stops itself, is fundamental for short-term survival, preventing runaway cell growth. And P16 is a key marker, especially with age, and even a tool for eliminating these cells in experiments.
You've got it. It's a testament to the elegant complexity of cellular defense. It really highlights the multi-layered defenses our cells have evolved.
It really does. An ancient and highly effective program built right into our biology.
Right.
We've established that senescent cells are growth arrested. They no longer divide. But, and this is a crucial point you made earlier, this doesn't mean they're dormant or inert, like a retired worker just sitting there doing nothing.
Far from it.
In fact, they are far from idle. They remain metabolically highly active with altered metabolism, and they adopt a truly distinctive secretory profile. This brings us back to the senescence-associated secretory phenotype, or SASP.
Yes. It's truly remarkable in its complexity and its potency. It isn't just a handful of molecules. We're talking literally dozens, maybe even hundreds, of distinct factors that these senescent cells actively and deliberately spew out into their surroundings.
Hundreds. Still sounds like a lot.
It is, and this isn't random leakage. It's an organized, highly regulated release of a specific complex cocktail of signaling molecules. It's their primary means of communication once they've stopped dividing.
Okay, so what's in this cocktail? What are they broadcasting?
Well, to truly unpack its composition, it includes several key categories. First, potent pro-inflammatory cytokines. Things like interleukin-6, IL-6, and interleukin-8, IL-8. These act like alarm bells, signaling inflammation to neighboring cells and tissues, kicking off a local inflammatory response.
Inflammation signals. Got it.
Second, chemokines. These are different. They're like chemical breadcrumbs or navigational beacons that specifically direct the migration of various immune cells to that particular location. They act as homing signals.
Calling specific cells to the scene.
Exactly. Third, various growth factors like hepatocyte growth factor, HGF, or vascular endothelial growth factor, VEGF. These can stimulate the growth, proliferation, or differentiation of other cell types, often in unexpected or context-dependent ways. They can even influence blood vessel formation.
Growth signals, too. Interesting mix.
Fourth, and critically important, are proteases, particularly matrix metalloproteinoses, MMPs, like MMP1 or MMP3. These are powerful enzymes capable of breaking down the extracellular matrix, or ECM.
The scaffolding between cells.
Precisely. The scaffolding and structural components that hold tissues together and provide cues to cells. This breakdown by MMPs can dramatically alter tissue architecture, for better or worse.
Remodeling the neighborhood.
Right. And finally, these cells also actively release extracellular vesicles, including things called exosomes. These are tiny sacs, like miniature delivery trucks, filled with molecular cargo proteins, lipids, various types of RNA. They are capable of influencing distant cells by delivering this cargo, effectively extending the reach of the senescent cell far beyond its immediate vicinity.
Wow. So it's not just a single ingredient, but a whole symphony, or maybe a cacophony, of molecules being broadcast, profoundly altering the local tissue ecosystem. Think of it like a cellular radio station, constantly transmitting a complex set of signals into its neighborhood. This complex mix must have diverse impacts. Does it always create chaos, or can it actually be beneficial?
That's the core of the paradox, isn't it? The impact of the SSP on the local tissue ecosystem is critically dependent on the context specifically, whether the senescence is acute and transient or chronic and persistent.
Acute versus chronic again.
Exactly. In acute contexts, such as wound healing, tissue repair, or even during early developmental stages. The SASP acts as a powerful healer and coordinator.
Good.
SASP factors help to actively coordinate regeneration. They orchestrate the entire tissue repair process, guiding it towards a successful and complete outcome.
How does it do that?
Well, it includes the efficient recruitment of immune cells to the injury site, as we mentioned. These immune cells, drawn by chemokines and cytokines in the SASP, act like that highly efficient cleanup crew. They clear away damaged cells, cellular debris, any pathogens that might be present. This immune surveillance and clearance is absolutely essential for proper healing and preventing infection.
Okay.
Furthermore, the SAP can directly signal to nearby stem or progenitor cells, encouraging them to proliferate and differentiate, effectively rebuilding the damaged tissue by generating new, healthy cells.
Stimulating the repair crew.
Right. Consider specific examples. When you get a skin cut, transient senescent cells quickly appear at the site. They secrete SSP molecules that attract specific immune cells, like macrophages, which are crucial for clearing damaged tissue and secreting growth factors that promote repair.
Ah, so they orchestrate the whole thing.
They play a key role. Similarly, in liver repair following acute injury, senescent cells and their SASP play a vital role in aiding healing and facilitating fibrosis resolution. That's the breakdown of scar tissue, which is important to prevent excessive scarring and help maintain liver function.
Preventing long-term damage.
Exactly. Moreover, some SASP molecules can even act back on the senescent cell itself in an autocrine manner, reinforcing its own growth arrest and ensuring it stays out of the cell cycle. Keeps the damaged cell locked down.
Self-reinforcing stop signal.
And importantly, the SASP also plays a crucial role in initial immune surveillance by attracting cytotoxic immune cells, natural killer cells, and cytotoxic T lymphocytes. These are immune cells specifically designed to identify and eliminate precancerous senescent cells.
The cellular assassin.
Right. This is a crucial early defense mechanism against tumor formation, a kind of cellular quality control.
That sounds incredibly beneficial, almost like a cellular orchestra conductor for healing. So where does the two-edged sword aspect come back in for the SSP itself? When does this beneficial acute response turn from a helper to a hinderer, from a conductor to a disruptor?
The problem arises, as we've hinted, in chronic contexts. When senescent cells persist and are not efficiently cleared from the tissue, when these cells accumulate and remain for prolonged periods, their continuous SSP becomes overtly detrimental. It transitions from beneficial to pathological.
The chronic state is the bad state.
Generally, yes. This persistent SAP creates a chronic, low-grade, pro-inflammatory, tissue-degrading environment. This is precisely what contributes to inflammaging, where chronic sterile inflammation becomes a fundamental driver of the aging process itself.
And this does actual damage.
Oh, yes. This isn't just an annoyance. It actively disrupts normal tissue structure, compromising the intricate architecture required for proper organ function. It impairs the function of resident stem cells, meaning they can't repair and regenerate tissues as effectively. Overall, the regenerative capacity of the tissue is significantly diminished.
How does it cause the damage? What are the mechanisms?
The mechanisms of damage are varied and, frankly, insidious. The proteases in the SAP, like the MMPs we mentioned, continuously break down the surrounding extracellular matrix, weakening the tissue, contributing to its deterioration.
Chewing up the foundations.
Pretty much. The cytokines, like IL-6 and IL-8, spread inflammation, not just to neighboring cells, but potentially systemically, contributing to a body-wide inflammatory burden. And crucially, a truly insidious aspect is paracrine senescence.
Paracrine senescence. What's that?
That's where SASP factors released by one senescent cell can actually propagate senescence to neighboring, previously healthy cells.
Spreading the senescence, like a contagion.
Sort of like a contagion, yes. It creates a domino effect, turning a localized problem into a widespread tissue burden, essentially spreading the bad neighborhood effect.
Wow, that's worrying.
It is. Thus, in aged tissues or chronic disease sites, senescent cells truly act like rebellious cells that no longer divide but won't die. They're like rogue agents that have gone off mission. They aren't contributing by dividing and rebuilding, but they're actively causing profound harm by staying put and continuously secreting this inflammatory cocktail.
So they fuel disease.
They fuel a range of pathologies. It promotes fibrosis, that's the excessive and pathological formation of scar tissue that stiffens and impairs organ function, like in lung fibrosis or liver cirrhosis. It drives generalized degeneration across tissues, leading to organ decline. And paradoxically,
Another paradox.
yes, Despite their initial anti-cancer role in this chronic state, they can even support tumor growth within the microenvironment.
Wait, they help tumors grow? How?
Yeah, by creating an inflammatory niche that can actually promote cancer cell survival. Angiogenesis, that's new blood vessel formation that tumors need to grow, and even metastasis, the spread of cancer.
That's the opposite of their original job.
It's a complete reversal, driven by the chronic context. There's also a fascinating, even more paradoxical immune effect. Early in senescence, some SSP components, maybe certain interleukins or growth factors, can transiently induce an immunosuppressive niche right around the senescent cell.
Suppressing the immune system locally. Why?
Well, the thinking is this might initially serve a protective purpose, maybe transiently dampening excessive immune activation to prevent overshooting the repair process and causing collateral damage to nearby healthy tissue, a bit of local control.
Okay, makes sense. But there's a catch.
There's a significant catch. This transient immunosuppression, if it persists because the senescent cells aren't cleared, can inadvertently allow emerging cancer cells to evade immune detection and elimination.
It shields them.
It can give those nascent cancer cells a critical window to establish themselves and grow into overt tumors, a dangerous unintended consequence of the persistent senescent state.
Wow.
And over time, the SEC composition itself often shifts, becoming even more overtly pro-inflammatory, solidifying its role in pathology. This chronic state contributes to widespread systemic diseases like insulin resistance, atherosclerosis, osteoarthritis, neurodegeneration, and numerous other age-related conditions. Therefore, senescent cells are increasingly considered key engines of aging and chronic illness, not merely a consequence or a bystander.
That's a truly intricate system, a delicate balance where context is everything. But you mentioned something else about the SASP earlier, its heterogeneity. Does that mean the cocktail isn't always the same, or are there universal ingredients?
That's a critical point for understanding and effectively targeting senescence. The SASP is not one size fits all. Its precise composition is highly variable, making it incredibly heterogeneous and context-dependent.
What determines the mix?
What a senescent cell secretes depends significantly on several factors. The cell type that became senescent in the first place, for example, a fibroblast, which is a connective tissue cell, versus an immune cell, versus an endothelial cell lining a blood vessel. They all have different jobs normally, and their SAP reflects that.
Okay, cell type matters. What else?
The specific trigger that induced senescence also matters. Was it telomere shortening, oncogene activation, oxidative stress? Each trigger can lead to a subtly different SASP profile.
So the why influences the what they secrete.
Exactly. Pioneering studies by researchers like Capet and colleagues really reviewed this remarkable complexity. They showed that when you look at different types of senescent cells, only a subset of SAP factors is actually shared across them.
Ah, so not everything is different, but a lot is.
Right. For example, senescent fibroblasts might have some common SAP molecules with senescent epithelial cells, which line surfaces, but many other secreted molecules will be distinctly different between them. In fact, large-scale proteomics, the comprehensive study of all proteins and secretome analyses, have revealed that out of hundreds of potential AFP factors, only approximately 19 factors seem to be universally elevated in SASPs across diverse cell types and various senescence inducers.
Only 19 universal ones out of potentially hundreds.
Roughly, yes. It highlights the vast diversity.
So it's not a single universal broadcast, but more like each senescent cell population is its own unique radio station, broadcasting a tailored mix of signals based on its origin and its experience.
Precisely. That's a great way to put it. This means that each senescent cell population secretes a unique mix of signals, a unique radio broadcast tailored by its original cell identity and the specific stress context it experienced.
And this heterogeneity, it must have consequences.
Absolutely. This SAP heterogeneity might very well explain why senescent cells in different organs or tissues, say senescent immune cells versus senescent fibroblasts, can have profoundly distinct influences on their surroundings and contribute to different pathologies within the same organism.
So one type might drive lung fibrosis, another arthritis?
That's the idea, yes, partly due to the specific SASP each one emits. This also highlights a significant challenge for researchers. Why a single SASP factor, like IL-6, which is often measured, is usually insufficient as a lone biomarker of senescence or as a single therapeutic target.
You need more than one signal to be sure.
Exactly. To accurately gauge senescent cell burden and activity in the body, panels of SASP factors are often needed. They're like creating a more comprehensive molecular fingerprint, needing a full criminal profile rather than just one identifying feature.
Makes sense.
This incredible diversity raises an important question about precision in intervention. If we are to effectively target senescence, we must understand this remarkable variability. It's not a monolithic enemy. It's a highly adaptable, context-specific cellular state demanding nuanced approaches.
That makes perfect sense. Okay, let's do a quick recap of this incredibly detailed chapter on the SASP. We've learned it's a remarkably complex, secreted cocktail.
Dozens, maybe hundreds of factors?
Cytokines, chemokines, proteases, growth factors, the works. And it can both aid acute healing, recruiting immune cells, promoting repair, suppressing early tumors.
Good AZSP.
But when it becomes chronic, because the senescent cells stick around, it drives chronic inflammation and inflammaging tissue degeneration, and can even spread senescence to neighbors.
Pterocrine senescence, yes. Fueling fibrosis, degeneration, even helping tumors sometimes.
And we learned about the tricky immune effects, sometimes suppressive, sometimes inflammatory. And crucially, this SASP is super heterogeneous.
Highly variable. Depends on the cell type, the trigger. Only about 19 universal factors.
Which makes biomarkers tricky and highlights the need for tailored therapies like senolytics to clear the cells or xenomorphics to calm the SASP.
Exactly. It's a cellular program with far-reaching systemic consequences, where the balance between acute benefit and chronic detriment is everything. This delicate balance, this tissue ecology, is precisely why xenotherapeutics are being developed.
Okay, let's unpack this even further. We've established that senescent cells accumulate with age, and their chronic SASP seems to drive a lot of age-related problems. But the really big question, the profound question is, are they actually causing aging? Or are they just, passengers on the aging journey, accumulating as a consequence of time and damage, but not actively driving the process itself?
That was the fundamental question, yes. Correlation versus causation.
Exactly. And this is where the world of mouse studies provided truly landmark insights, fundamentally shifting our understanding from mere correlation to direct, undeniable causation. This was a huge paradigm shift in aging research, wasn't it?
Oh, absolutely huge. These landmark mouse experiments provided direct, causal evidence that fundamentally transformed the field. It wasn't just observing, oh, look, older mice have more senescent cells. It was about intervention. By systematically removing these specific cells, scientists demonstrated a direct, measurable influence on the aging process itself.
Okay, paint the picture for us. Describe one of these groundbreaking experiments. You mentioned visualizing it earlier.
Right. Let me guide you to visualize the profound results of one such key experiment. This was often done with mice that had reached the equivalent of an elderly human, maybe around 22 months old, which is quite aged for a standard lab mouse.
Okay, elderly mice.
Now imagine two of these elderly mice side by side, perhaps on a lab bench under observation. On your left, envision the untreated control mouse. This mouse exhibits the clear, unmistakable hallmarks of advanced age for its species.
What would that look like?
Well, perhaps its fur is dull, maybe patchy, thinning in places. Its posture might be somewhat hunched, indicating frailty. Its movements are noticeably slower, less coordinated, signifying a general decline in physical activity and muscle tone. You might even observe signs of common age-related issues like cataracts clouding its eyes or perhaps visible masses beneath the skin. It looks unmistakably old and, frankly, unwell.
Okay, typical signs of aging.
Yes. Now, shift your gaze to the mouse on the right, and this is the truly striking, almost unbelievable part. This mouse, same age, same background, but it underwent a targeted intervention, and it appears remarkably, profoundly different.
How different?
For its age, it looks significantly healthier, more vibrant, and undeniably more youthful. Its coat might be glossy and full. Its posture is more erect and robust. It moves with much greater vitality, activity, and coordination. There's a noticeable vigor, a truly youthful appearance for its chronological age, especially when you compare it directly to its untreated littermate on the left. This dramatic visual contrast alone provided a powerful initial indication that something profound and unprecedented had occurred.
That visual difference alone is incredibly compelling. It's like looking at two different generations of mice, almost. How exactly did they achieve this dramatic, almost age-reversing outcome in the treated mouse? What was the intervention?
This brings us right back to what we discussed in Chapter 2, connecting the dots of the science beautifully. This dramatic outcome was achieved through the ingenious use of sophisticated genetic engineering. Specifically, researchers utilized those transgenic mouse models we touched upon, like the INK attack system.
Right, using the P16 marker.
Exactly. As you'll recall, P16-INK4A is that broad and powerful biomarker that robustly increases with age, and it's a key enforcer of senescence. In these cleverly engineered mice, the natural P16 promoter, the genetic switch that turns on P16 when a cell becomes senescent, was precisely linked to that suicide gene.
The one activated by a drug.
Right. A gene that, when activated by a specific, often orally administered drug, a harmless chemical compound known to induce dimerization, would trigger the selective elimination, apoptosis, of only those cells that were expressing P16, meaning the senescent cells.
And they do this continuously.
The key here is that these P16-INK4A positive senescent cells were periodically cleared. This wasn't a one-time injection or treatment. It typically involved ongoing systematic removal of these cells as they accumulated over the mouse's later life, maybe treatment twice a week or once every 2 weeks, reflecting a continuous intervention strategy.
Okay.
This method provided an unparalleled level of specificity and control, allowing researchers to isolate the effects of senescent cells like never before.
And what were the precise measurable outcomes of this periodic clearing? What specific improvements did they observe beyond just the striking visual appearance of youth? What actual physiological changes occurred?
The outcomes were truly profound and went far, far beyond just the cosmetic appearance. The mice that had their senescent cells periodically cleared showed significantly delayed age-related deterioration across multiple, diverse physiological systems.
Not just skin deep.
Not at all. We're talking a slower onset or even prevention of many of the typical signs of aging that we associate with functional decline. For instance, studies reported improved kidney function, better cardiac performance like resistance to stress, increased muscle strength and endurance, reduced accumulation of fat, especially the unhealthy visceral fat, and even maintenance of better cognitive abilities in some models, suggesting positive effects on brain health.
Wow, systemic benefits.
Yes, their skin appeared healthier, they developed fewer cataracts, their overall physiological resilience, their ability to withstand stress and recover was notably enhanced. They were just healthier old mice.
And did they live longer?
Crucially, the focus and the most significant outcome reported consistently was an extended health span. This distinction is vital and central to the entire field of geroscience and aging research.
Health span again, not just lifespan.
Exactly. It's not simply about making an organism live longer chronologically, although some studies using senolytics or genetic clearance have also shown modest increases in overall lifespan, maybe 15–30% in some cases. But the primary, more consistent, and arguably more important finding is about extending the period of life spent in good health, free of debilitating disease and disability.
Quality over just quantity.
Precisely. These mice weren't just living longer. They were living better for significantly longer, maintaining a higher quality of life into what would otherwise be their frail, elderly years. This directly aligns with the broader goal of xenotherapeutics that we introduced earlier to extend health span.
And this proved causation.
This experiment, and others like it that followed, using different genetic models or pharmacological senolytic compounds, provided direct causal evidence. It wasn't just observing a correlation, not merely an association. By systematically removing these specific cells, scientists demonstrated a direct, active influence on the aging process itself. It truly changed the game.
Smoking gun.
You could say that. It strongly suggests that if clearing a specific cell type can have such profound, multi-systemic effects on health span in mice, then similar approaches hold immense implications for human aging and the treatment of age-related diseases. It raises that profound question. Could we one day, with similar or perhaps even better precision, re-engineer aspects of human aging?
So to reiterate, this landmark mouse study delivered profound findings. Directly clearing senescent cells, often tracked by P16, significantly delayed age-related decline across many systems and extended their healthy lifespan.
A remarkable outcome.
This truly moved senescent cells from being mere markers or correlates of aging to proven active and potentially modifiable causal drivers of the aging process itself.
It confirmed a hypothesis that had been building for decades, really validating senescent cells as a critical target and opening up entirely new avenues for therapeutic intervention aimed at promoting healthy aging. #outro.
So what does this all mean for us? We've journeyed from the microscopic world of cellular biology, the intricate dance of proteins like P16 and P21, the complex signals of the SASP, all the way to the macroscopic implications for organismal health and aging.
It's quite a journey.
We've seen how cellular senescence, a program ingeniously designed to protect us in our youth stopping cancer, helping wounds heal, can paradoxically transform into a silent saboteur over time, relentlessly driving many of the very conditions we associate with aging through that chronic inflammation and tissue disruption.
The double-edged sword in action.
And critically, those groundbreaking mouse studies didn't just show a correlation, they established a causal link. They provided direct evidence that targeting these cells can fundamentally alter the aging trajectory, at least in mammals.
A crucial step forward.
So it's no longer just about understanding aging as this inevitable decline. It's about potentially re-engineering it, influencing the very biological processes that underpin that decline.
Indeed. This journey, understanding senescence moving from a cellular emergency brake to a systemic burden, it profoundly alters our perception of aging itself. It shifts it, potentially, from an inevitable, unmodifiable process to one that, at least in part, can be understood and perhaps actively influenced.
A paradigm shift in thinking.
Absolutely. If, as the evidence strongly suggests, we can develop ways to specifically target and modulate these cells or their signals safely and effectively in humans, well, the implications reach far beyond conventional medicine.
Oh, so?
They touch on our very definition of what it means to age, what it means to live a full and healthy life. It pushes us to reconsider the limits we currently accept. And this raises an important question, perhaps a philosophical one, for you, for all of us to consider. If we possess the knowledge and the nascent tools, and they're still nascent for humans, we must stress, but if we develop the capacity to intervene in a process as fundamental as cellular aging, a process that has shaped all complex life on Earth for eons, what new responsibilities does that unlock? What complex ethical considerations arise? And what profound philosophical questions might that capacity unlock for humanity as we look towards the future? It's a future that's rapidly moving from the realm of science fiction towards scientific possibility.
A future where aging itself might be treatable, a truly profound thought to end on.